Hello, and welcome to the ACR Journals on Air podcast. I am your host, Vicky Shamigam. Every budding rheumatologist has seen the episode of House in which the interns present a case of complex systemic disease characterized by a dysregulated immune response leading to widespread inflammation and end organ damage, and postulate that the patient might have lupus. Dr. House rebuffs them with, "It's never lupus," leaving the interns and the audience questioning, how can he be so sure? What if there was a better way to measure the biological pathways leading to tissue inflammation in lupus and other autoimmune diseases? And what if NETs and low-density granulocytes have the answer? The manuscript we will discuss today is Exploring the Role of Neutrophil Extracellular Traps in Systemic Lupus Erythematosus, a clinical case study and comprehensive review. It was published in Arthritis and Rheumatology, and a link to the manuscript and disclosures are in our show notes from today. It is truly an honor to be joined today by the author of this manuscript, Dr. Mariana Kaplan. Dr. Kaplan is the chief of the Systemic Autoimmunity Branch at the National Institute of Arthritis, Musculoskeletal, and Skin Diseases, or NIAMS, at NIH, and she is also Deputy Scientific Director of the Intramural Program. Dr. Kaplan obtained her medical degree at the National Autonomous University of Mexico and did her internal medicine residency at the National Institute of Medical Sciences and Nutrition in Mexico City. She then completed a rheumatology fellowship and postdoctoral training at the University of Michigan, where she was a faculty member for 15 years and an active member of the Multidisciplinary Lupus Clinic. Now, in addition to her research activities at NIH, Dr. Kaplan is an active clinician and teacher, seeing lupus patients through the NIH Clinical Research Center, where she's involved in developing clinical trials for patients with autoimmune diseases. Dr. Kaplan has received numerous awards throughout her career, including the Henry Kunkel Young Investigator Award, the Edmund L. Du Bois Memorial Lectureship, and the Distinguished Basic and Translational Investigator Award from the ACR, as well as the Evelyn V. Hess Award from the Lupus Foundation of America. She serves on the editorial board of several journals, and in 2021, she was elected to the National Academy of Medicine for her seminal contributions to autoimmune diseases, atherosclerosis, and immune-mediated vasculopathy. Welcome to the show, Mariana.
Well, thank you very much, Vicky. I'm really delighted to be here, and thank you for the kind invitation to participate.
So this paper, like several that we've covered on this podcast, is a review article, and it tackles a problem we've known about in lupus for a long while. Making a diagnosis is challenging, and lupus carries a risk of accelerated cardiovascular disease. But until recently, we've not really understood the biologic pathways that make this happen. I wondered if you could summarize the case for our listeners and make the case for why this was important to study.
Certainly. So I'm going to start by telling you a little bit about this patient who is a 31-year-old woman who presented to the rheumatology clinic with a 6-month history of fatigue, joint pains, fevers, and a facial rash that was increased or exacerbated by sun exposure. And she had first noticed these symptoms after the resolution of an upper respiratory tract infection. She also had non-painful recurrent oral ulcers, Raynaud's, and hair loss that had been present for about 3 months. And she had taken acetaminophen for joint pain, had tried to stay away from the sun, but her symptoms had persisted. So on physical exam, she was found to have an erythematous macular rash that spared the nasolabial folds, oral ulcers, tenderness and swelling in multiple joints, as well as evidence of synovitis in her hands and knees. And on laboratory tests, she was found to be leukopenic, lymphopenic. She had also mild neutropenia, elevated inflammatory markers. And then she was found to have high titers of ANA on a homogeneous pattern, high titers of anti-double-strand DNA, anti-Rho antibodies, and decreased complement levels. She had normal kidney function. However, she was found to have cardiometabolic dysfunction characterized by low levels of high-density lipoprotein, HDL, elevated LDL, and total cholesterol. And when she underwent tests for subclinical vascular disease, a carotid ultrasound was shown to have evidence of increased carotid intimal media thickness bilaterally, And when we went further more on the research side, she was found not only to have low HDL, but this HDL had impaired function. And we also found evidence that on peripheral blood, on the circulation, she had enhanced levels of circulating nets and enhanced levels of subset of neutrophils called low density granulocytes. So, given this clinical presentation, we diagnosed the patient with lupus and with preclinical atherosclerosis and dyslipidemia. We initiated an antimalarial as well as a statin and low dose prednisone. And we advised her to stay out of the sun and use sunscreen, take vitamin D, and get her vaccinations and other general preventive measures for lupus comorbidities. So I think this case really highlights the classical presentation of lupus, as we know, characterized by a constellation of constitutional symptoms, mucocutaneous manifestations, multi-system or multi-organ involvement, and the presence of cardiometabolic dysfunction, and really highlighting that lupus represents one of the most complex autoimmune conditions, really a syndrome, characterized by significant heterogeneity in clinical manifestations, but also this association with accelerated cardiovascular damage and cardiovascular disease that has been a long-standing enigma. And so this review really had tried to aim to bridge the gaps in understanding the role of neutrophils and neutrophil extracellular traps in the pathophysiology of lupus, using a case to integrate recent advancements in the field.
That's a wonderful introduction. So let's dive a little deeper into the immunology. Lupus involves this complex interplay between the innate and adaptive immune responses. What role do the neutrophils play in that?
Yes, no, thank you. So for many years, the dogma was that lupus was primarily a disease of the adaptive immune system, where both autoantibodies, B cell dysregulation, and T cells were the major players in inducing breaks intolerance, downstream immune dysregulation, and organ damage. And of course, the adaptive immune system is extremely important in lupus. But over the last couple of decades, we have come to the realization that components of the innate immune system, which is the first response, the first line of defense against infections and other danger signals, is also playing critical roles in lupus pathogenesis. And within the innate immune system, our work and the work of others has emphasized that neutrophils are indeed key players in the pathogenesis of lupus. And this may go through the initial stages of, or even before patients are diagnosed with lupus, inducing loss of tolerance, initiating autoimmunity, but also moving forward through the lifespan of an individual, perpetuating the disease once autoimmunity develops, and also playing crucial roles in inducing organ damage and cardiovascular disease.
Yeah, that's a brilliant introduction. I'm going to jump you forward in your article now, because in figure 1, you have this wonderful graphic that shows the complex web of inflammation that neutrophils and neutrophil extracellular traps weave in lupus. Can you walk our listeners through what those graphics show?
Yes. So what we're trying to show in this graphic is that there are a number of stimuli that our bodies are constantly exposed to, microbes, but also other inflammatory stimuli that may be what we call sterile, not induced by infections, but by other danger signals, that have the ability to induce these structures called nets. And once the nets are formed, they externalize a number of what we call alarmins, which are molecules that activate the immune system, but also a lot of different types of nucleic acids, DNA, genomic DNA, mitochondrial DNA, RNAs, that may be externalized also in a more oxidized manner, because neutrophils are highly oxidative cells. And so these nucleic acids become oxidized and are extruded. They can also externalize histones that are part of the nucleus. And when these structures are externalized, they have the ability to activate many immune and also stromal cells in tissues that can promote inflammation and tissue damage. And the idea of this schematic is also to highlight that if net formation is enhanced and or the clearance of these nets is impaired, then this enhanced half-life of the nets can lead to their persistence in tissues, in the circulation, and exacerbate the deleterious roles of these structures regarding activating the immune system, inflammation, or promoting organ damage.
So I wonder if we could take a little detour for a moment and talk about the concept of netosis. What is it? Why does it happen? And what are the triggers in patients with autoimmune diseases and particularly lupus?
Yes. So the formation of nets or netosis was described for the first time about 20 years ago. And before then, we knew that neutrophils basically get rid of microbes or encounter microbes through a number of of strategies that were primarily what we call degranulation, the release of granules to the extracellular space, or phagocytosis of certain microbes, the formation of reactive oxygen species. And then 20 years ago, there was this description of this new antimicrobial strategy whereby the neutrophil extrudes the nuclear material, DNA, RNA, histones, bound to proteins present in the neutrophil granules, myeloperoxidase, proteinase 3, and others, in this meshwork that really looks like a spider web into the extracellular space. And these structures were called neutrophil extracellular traps or nets, and the mechanism by which the neutrophils die by forming the nets, netosis. And it became clear that this is a crucial antimicrobial strategy, but that there are also, as I mentioned, sterile inflammatory stimuli, not only microbes, that have the ability to induce net formation. And among them, many are highly prevalent in individuals either at risk or who have already developed autoimmune diseases such as lupus. And I may mention then that autoantibodies, immune complexes, certain cytokines, but even platelets or various crystals such as cholesterol crystals or uric acid crystals or medications that have been associated with the induction of autoimmunity also have an enhanced ability to form these nets.
And so tell us about what's in the nets, and in particular, citrullinated histones are the hallmark of nets. What are they and why does this happen?
Yes. So as we know, the nucleus, besides nucleic acids, also carries these proteins called histones and DNA form the nucleosomes, which are integral parts of the nuclear structure. And during the process of net formation, inside the nucleus, there is the activation of this enzyme called peptidyl arginine deaminase 4 or PAD4, and neutrophils are the cells expressing PAD4. And PAD4, when activated as nets get induced, citrullinates histones. And this citrulination of histones leads to the expansion of the chromatin. And therefore, this is a very early event that is crucial for the activation of the process of net formation. And so without histone citrulination, it is believed that many forms of nets may not be possible. And we use that also as a sort of a marker that the neutrophil has engaged in the process of net formation.
And so can you help us understand how genetic risk factors, particularly genetic polymorphisms that confer risk of lupus, also impact net formation?
Yes, thank you. So this is a very interesting field that is evolving. But we now know that many of the genetic polymorphisms that are associated with an enhanced risk to develop autoimmunity, and in particular lupus, are also associated with an enhanced risk for neutrophils or an enhanced ability for neutrophils to develop nets. And for example, polymorphism of IRF5, STAT4, even PAD4 or PTPN22 have been associated with enhanced activity of PAD4 or in other for other less clear mechanisms, an enhanced ability of neutrophils to form nets. And we may think, well, it's a chicken or egg question. Could it be that these people already are at risk for lupus and they're formed because of that, they're forming nets? But there have been studies even in people that have not developed lupus but carry some of these genetic polymorphisms like RIF5. And it has become evident that people that carry the IRF5 polymorphism for lupus but have not developed lupus are already forming more nets than people that do not have this genetic risk allele. And so it appears that one of the mechanisms by which these genetic risk alleles promote lupus could be through enhanced net formation.
So that's really interesting. And as someone who's looked after lots of people living with lupus, the unpredictability of flares of lupus is a real challenge. Is the current thinking that flares in established lupus are also a net-driven process?
So I think more research is needed. And as we know, lupus is such a heterogeneous condition that we cannot say that, of course, in all patients, NETs are playing a role in lupus flares. But there is certainly evidence that is accumulating that suggests that NET levels may go up during a lupus flare. We are doing longitudinal studies to assess if NET increases in NET levels may predict the flare. I think it's early for us to tell. But by releasing autoantigens and inflammatory mediators, we know that this net set off a cascade of immune responses that may exacerbate disease activity. And this understanding opens the door to exploring nets as biomarkers for predicting flares. This could be one of the links between infections and flares. We have known for many years that lupus patients may flare after, like the patient in this case, an upper respiratory tract infection or other type of infection. And certainly infections can trigger net formation. And so one possibility is that in an individual already at risk to develop autoimmunity or who has already an underlying autoimmune disease, these events that enhance net formation, such as flares, such as infections, or, for example, exposure to UV light, may be a mechanism that induces flares post these types of stimulation.
Yeah, that makes a lot of sense. So let's talk now about these low density granulocytes, or LDGs. What are these cells and why are they important in lupus?
So low-density granulocytes are considered a unique subset of neutrophils that are found at enhanced levels in patients with a number of autoimmune diseases, and particularly in lupus. And we call them low-density granulocytes because when we do a density gradient to isolate mononuclear cells such as lymphocytes and monocytes from blood, typically neutrophils go to the bottom of the tube next to the red blood cells because normal neutrophils have higher density than lymphocytes or monocytes. But in the case of low density granulocytes, we find them having lower density in the tube, and actually they have similar density to lymphocytes or monocytes. So therefore, we have named them low density granulocytes. But it's not only their density that is different, they actually have a number of differences when compared to normal density neutrophils from the same lupus patients or from healthy individuals. We consider that they are more pro-inflammatory. And one key characteristic of these cells is their exquisite ability to form nets, even without any stimulation, without presence of infection or other danger signals. And so we believe they may be important drivers of disease activity and of vascular damage.
It's amazing. So could LDGs be a surrogate biomarker for lupus disease activity?
So this is work in progress, and we are following patients longitudinally to see whether as the level of inflammation or as disease activity increases, we are seeing preceding that elevations in low-density granulocytes, or at the time of flare that these cells go up. We do know that they fluctuate as the disease with disease course. We just need to study more patients longitudinally to assess whether they could be used in the future as a biomarker for risk of lupus flare beyond what we already use, such as elevations in anti-double-stranded DNA or decreases in complement levels.
Yeah, that's really exciting and kind of anxious to see where that work lands. So tell us about the relationship between LDGs and atherosclerosis, and particularly accelerated atherosclerosis that we see in lupus.
Yeah, so this is an area we're very excited about because this is a big area of need in lupus. We know that for other chronic inflammatory diseases, cardiovascular risk has decreased over the last decade. So the risk for cardiovascular disease, and this may be the case for rheumatoid arthritis, inflammatory bowel disease, even psoriasis. However, in lupus, recent studies suggest that there have been no changes in this increased risk for premature cardiovascular disease. So there's something we're still missing of the mechanisms driving premature vascular damage in lupus. And what we have found is that when we do multivariate analysis adjusting for well-established risk factors for cardiovascular disease, tobacco, lipids, diabetes, being sedentary, etc., the presence and levels of these low-density granulocytes strongly associate with enhanced plaque formation in the coronary arteries of lupus patients and enhance inflammation of the blood vessels, such as the aorta or other smaller blood vessels. And we see clear association between the presence of these cells and cardiometabolic dysfunction in lupus patients. So we do think that because of their enhanced ability to form nets, which damage endothelial cells and accelerate the formation of atherosclerotic plaque, but also due through their enhanced pro-inflammatory phenotype, these lunacy granulocytes, this dual role may underscore their importance as both biomarkers for cardiovascular disease and also potential therapeutic targets in lupus.
Yeah, so it's a complex web. And it sounds like net formation is not just a driver of inflammation in lupus, but also increases exposure to autoantigens, and then that further increases inflammation. Can you share a bit more about how that process happens?
Yes. So what is really interesting about nets is that when they are extruded into the extracellular space, they externalize many autoantigens that are known to be key drivers of autoimmune responses in individuals either at risk or with well-established autoimmunity. And so, as I mentioned, nets, for example, carry a lot of DNA and modified DNA, but also modified RNA. And we know, and histones, and we know that patients with lupus, many develop autoantibodies to these autoantigens, double-stranded DNA, RNA, and histones. But they also carry, for example, high amounts of myeloperoxidase and proteinase 3 that are known to be key target of the immune system in ANCA vasculitis, and also citrulinated autoantigens because of this activation of PAD4 that I mentioned before. And so and this may be quite relevant in the case of rheumatoid arthritis. So nets are key carriers of modified autoantigens that are commonly targeted by the immune system in some of the most common systemic autoimmune diseases. And this is certainly the case for lupus patients.
So it's interesting. It makes me wonder whether the issue is just net production or whether there's also an issue of breakdown. So do you see a relationship with impaired net clearance also in lupus?
Yes. So about a third of lupus patients are known to have impairments in net clearance. And so what we see is a pretty profound imbalance between net formation that we know is enhanced, and part because it is driven by these low density granulocytes, but also because immune complexes and autoantibodies tend to trigger enhanced net formation. But also this third of patients that have impaired clearance may further promote this enhanced half-life of these structures in the circulation or in tissues. And the reasons why lupus patients have impaired clearance are probably multifactorial. Part may be genetically driven. We know that certain polymorphisms in DNAs or other nucleases limit the ability of these DNases to degrade the DNA present in the nets. We also know that there are autoantibodies that lupus patients make that may interfere with the ability of these nucleases to get rid of the DNA that is externalized in the nets. And we also know that macrophages and other phagocytic cells that may have the ability to phagocytose these structures may also be impairing their function. And so this inability to clear nets has been associated with an enhanced risk for lupus nephritis. And it is possible that in the future we will find other risk factors for organ damage associated to, or that nets, the impaired net for degradation serves as a risk factor for other complications in lupus beyond lupus nephritis.
Yeah, really, really fascinating. And I'm glad you brought up the issue of organ damage, because I've been wondering, how do the nets cause organ and tissue injury? In my mind, I'm sort of envisioning this spider's web getting caught in the glomerulus. Is that actually accurate, or is there a better metaphor for what's going on?
So this is a great metaphor you use. And in fact, it is true that we have been able to find these structures in various lupus organs, including the kidney and particularly the glomerulus, but also the interstitial space in the kidney. We also find them in lupus skin, especially in areas affected by lesions. We have seen them in placentas of lupus patients who develop preeclampsia. And so this seems to be not only just enhanced net levels in the circulation, in the blood, but these nets are able to get into the tissues. And we think that when that happens, there are pleiotropic roles of these nets that may lead to exacerbation of inflammation, somewhat creating a vicious cycle that activates other inflammatory signals that instead and that in turn lead to more net formation. But the process appears to be pleiotropic. Part may be direct damage to stromal cells, mesangial cells, endothelial cells, activation of fibroblasts that may lead to tissue fibrosis, activation of other immune cells present in organs, for example, macrophages that may further promote the activation of this very inflammatory pathway called the inflammasome and lead to enhanced production of IL-1 and IL-18. Nets can actually induce plasmacytoid dendritic cells to synthesize higher levels of type 1 interferons. And we know that type 1 interferons are very important cytokines in lupus pathogenesis. And so this is really a very inflammatory cycle that is triggered by these structures when they are produced at enhanced levels are not properly cleared in lupus patients.
Yeah, that's a great explanation. And at the beginning of the episode and in the beginning of this manuscript, you talk about sort of research testing that was done on the specific patient that presented in this case. But testing for NETs and LDGs is not currently commercially available. What would need to happen to advance this science and bring it to clinical care?
Yes, that's an excellent question. Thank you. So I think we first need to validate whether low-density granulocytes and nets are indeed playing important roles in the disease. We have strong evidence from preclinical models and some proof of concept clinical trials that this indeed may be the case, but this needs to be expanded into larger cohorts and clinical trials and continuing natural history studies to assess the role as biomarkers of disease, predictors of flare, predictors of severity and risk for organ damage, as well as vascular disease, where we already have some strong hints that this may be the case. And then if this is validated, and hopefully in the future we can use them as for prognostic value or for diagnosis, we will need to establish tests that can be easily implemented in clinical labs so that they can be incorporated into clinical trials or even in the future clinical practice.
Yeah. And I'm going to ask you to speculate now, because if studies continue to support this hypothesis that nets and netosis are important drivers of both disease and also flare in lupus, could this open avenues for new therapeutic targets, and what might those be?
Yes, so absolutely. We think that targeting nets could provide new therapeutic avenues for lupus. But again, more work is needed. And we have already been performing some proof of concept studies at the NIH. And we have tested some therapies, some which are actually repurposed drugs, but we're also very excited to be in the process of hopefully testing some new drugs for therapeutic target. And I can tell you, for example, that we have found that drugs such as JAK inhibitors have the ability to block net formation and stabilize or decrease the number of low-density granulocytes with some positive effects on research tests on blood vessel damage or vascular risk. We have also, we are currently testing the potential role of anifrolumab in an ongoing clinical trial to see whether blocking type 1 interferon pathway may lead also to decreased net formation and have a positive effect in vascular damage. And potential strategies could, for example, could include the use of PAD4 inhibitors of reactive oxygen species generation, or other strategies that limit the damaging effect of neutrophils, but that do not totally blunt the very important antimicrobial role that neutrophils play. So if we could selectively inhibit this dysregulated net formation without modifying other functions that neutrophils have that could make an individual more prone to infections, this would really be the ideal way by which we could target dysregulated net formation and inflammatory pathways without putting individuals at higher risk for infections. So this is really ripe for further research and development.
It's great to hear you describe this, because it really emphasizes this concept that there's a matrix going on and lots of different nodes where potential therapeutic targets could intervene, or the body's own immune system can alter levels of these nets.
Thank you.
So I want to thank you, Mariana, for sharing all those insights into the world of NETs. Our listeners are also interested to know a bit about the stories behind the science, and I wondered if you'd be kind enough to share some background about your career. You began your career in Mexico and then moved to Michigan for fellowship, very cold part of the country. What prompted you to move so early in your career, and why did you pick rheumatology?
Yes, thanks. Yeah, so certainly wasn't the weather, as you say, when I moved to Michigan. So my journey, as we mentioned, began in Mexico, and I really developed there a passion for immunology and for, more particularly, for autoimmunity. And this really started for me when I was in medical school, when I encountered for the first time some young patients with lupus, and was really struck by first why this disease affects primarily young women, and how many organs and systems are involved, and really this development of what we call loss of tolerance to self. And so I decided pretty early during my medical studies that I wanted to become a rheumatologist, and I was really interested in studying autoimmunity and in particular lupus. So my move to Michigan was really motivated by the opportunity to combine and learn bench research with clinical rheumatology. So I had been exposed to really great clinical training, but I wanted to test whether bench research was for me. And that's why I joined the University of Michigan for my training. And then as a faculty member at the University of Michigan, I was able to continue my research after my training and really be fully focused on autoimmunity and lupus.
And so, as a faculty member at the University of Michigan, you pursued a pretty traditional physician scientist career pathway. Would you share with our listeners how did you win your first grant? And what was it like as you grew from a junior faculty to an established scientist?
No, thank you. So I think my very, very first grant was actually from the Life Sciences Research Institute, which I'll be always grateful to because they gave me this postdoctoral grant to continue my research. And then I was very fortunate to get at the time what is called a KO8 grant, which is a mentor grant for junior faculty. I was an assistant professor at the time. And that led me to really start pursuing some of my own lines of research related to, how the innate and adaptive immune system are involved in the pathogenesis of lupus, expand my lab with these funds, as well as with additional funds I was able to get from the NIH and from a number of organizations such as at the time the Lupus Research Institute, now the Lupus Research Alliance or the Lupus Foundation of America, and start my self-training and people to get interested in studying autoimmunity and lupus. And so it's really been a privilege and a pleasure to be able to try to teach what I've learned from others and to try to expand the field of lupus research by attracting new people into the field.
Yeah, that's great to hear. Tell us a little bit about being recruited to the NIH in 2013. What enticed you to make that move? Did the US government offer you all the neutrophils in the world?
Yeah. So, as you know, NIAMS have been, a great center for lupus research, and some of the, initial studies on, for example, the use of cyclophosphamide in lupus and other therapeutics were done at the NIH. And so there had been a period at the time where not a whole lot of lupus research was going on at the time I was recruited. And so there was a wish to revitalize the lupus program. And I thought that was a really interesting and attractive possibility for me, and that the NIH, of course, would offer unparalleled resources to advance lupus research, focusing on high risk, high reward science and translational approaches. And so I was, although while I love my time at Michigan and I have the fondest memories, I thought this was, once in a lifetime opportunity. And I was really excited to move to the NIH 11 years ago.
So it's wonderful to hear, and it's exciting to watch what you've done since you've been here. So you're incredibly accomplished. You've won so many awards. I'm wondering if you could share with our listeners what it was like a few years ago being elected to the National Academy of Medicine. And for our listeners, could you share a little bit of background about the organization, what they do, and what do you have to do as a member of that academy?
Yeah, thank you. So, being selected to the National Academy of Medicine was really a humbling recognition and honor, and a milestone for me, reflecting recognition for my contributions to immunology, to lupus research, to mechanisms of vascular damage. And so I feel extremely honored to be part of this group of outstanding physician scientists. And as a whole, the National Academy of Medicine fosters health policy development and scientific advancement. And so it asks its members to engage in initiatives to address pressing healthcare challenges. And so a lot, it's really about learning way beyond what our own fields are engaged in, but bigger picture of what is really, what are the challenges in health and disease, from, global warming to health disparities, but also really getting into understanding how the implications of the research that we all do into advancing healthcare. And so it's been a really exciting experience for me, learning more and more. And at times we may be asked to engage specifically in certain committees that want to address specific healthcare challenges, or to work with other disciplines to further understand a specific problem within medicine.
That's really amazing to hear, and I'm so glad that they have some rheumatologists in the academy. So, Mariana, what's next for you as a scientist? What are the experiments keeping you up at night?
Yeah, so thank you. So this is a great question, and I would say my major role or my major goal is I really want to try to translate our research findings into interventions that will improve patient outcomes, and ideally with a personalized approach. Lupus is an incredibly complex condition, and so the more we can personalize the treatment, the better our patients will do. But this is, as we know, complicated, and so a lot of what I do remains focused on try to unravel lupus immunopathogenesis with ongoing efforts to target innate immune dysregulation, and we are very focused on what the role of NETs and low-density granulocytes may play in that, and really trying to understand more broadly what role do neutrophils play in health and disease, and whether there are different types of neutrophils that may play different roles, because that may allow us to more specifically target the ones that we think are being the bad guys in the pathogenesis of autoimmune diseases. And so understanding their broader role across autoimmune diseases, we think could lead to perhaps paradigm shifting therapies.
Yeah, and that's amazing to hear, and makes me kind of think about potential for immunology to actually have paradigm shifting changes across other diseases too, particularly cardiovascular disease more broadly.
Oh, absolutely. And I think we are learning a lot from why premature cardiovascular disease develops in lupus. And I think this goes beyond lupus and even autoimmunity into understanding does all atherosclerosis develops the same way in the general population. And there may be subsets of people within what we call the general population that are at risk for cardiovascular disease, where mechanisms similar to what happens in lupus and other autoimmune diseases may apply. And therefore, preventive strategies or therapeutic strategies may also differ depending on whether this risk is more similar to the risk we see in an autoimmune patient or whether other pathways are involved. So I think what we learn in autoimmunity has implications far beyond these diseases.
Yeah, I completely agree. The immunome is the missing link. So I want to thank you, Mariana, for this wonderful discussion. I really appreciate you taking the time to chat with us today, and for particularly taking on this complex web of scientific discovery and spinning it into a beautiful story.
Oh, thank you very much, Vicky. I really appreciate the opportunity to talk about my work and my career. And thank you very much for all the great questions. And, thank you to the ACR also for allowing me to participate in this podcast.
So today, my thanks also go to fundamental scientists who dig deeply into the web of the immune system and help us understand the biology and drive fundamental discoveries that impact everyone living with and at risk for autoimmune disease. Thank you all for listening to this podcast. ACR Journals on Air is a production of the American College of Rheumatology. Please follow at ACRheum on social media, email us at podcast@rheumatology.org, and rate and subscribe to our podcast at acrjournalsonair.org or wherever you get your podcasts.